So thank you for inviting me here to Jogja. Thank you for this lovely flower. I come from Hawaii, or at least, so we know about flowers. But this is very special. I don't know. I've forgotten what you call them in Indonesia, but in Hawaii we call them lei. Yeah. So I first came to Indonesia in 1968, probably before many people here were born, and I have seen many changes. I don't know how many times I've come to Jogja, but I first started coming when the Rockefeller Foundation faculty was here. And then I joined the Rockefeller Foundation, and we brought a very important educational program integrating research into patient management called INCLEN. And then we conducted, with Dr. Ida's help, an important prospective cohort study. I'm sure Dr. Anders will tell you that one of the only ways we can learn about dengue is we need to study this disease over time, because with 4 viruses it's just too complicated. This is not measles, and it's not rubella. It's not something simple. So today I'm going to tell you about the status of dengue vaccines, where we are and where we are not, and the underlying theme of my talk is the word if, because you'll see there are many ifs, many problems that we still have to solve. Let me say, first of all, those of you who've been following the dengue vaccine development, a vaccine which was born by the Centers for Disease Control in the United States, so-called dengue chimera, dengue 2, dengue chimera, was licensed initially to a company called Invirigen, and then very recently has been purchased by Takeda Pharmaceuticals, a huge company with headquarters in the United States and in Europe and in Singapore. So here are the current vaccines that are in some stage of clinical development. And like most vaccines that we've ever developed in the world of virology, some are live attenuated, and as you can see, others are killed. So here are the 3 live attenuated vaccines that are in various stages of human development. Sanofi Pasteur has a dengue 1 through 4 spliced into the backbone of yellow fever. Takeda is a mutagenized dengue 2, 1, 3, and 4 chimera. And NIH has mutagenized dengues 1, 3, and 4, but has a dengue 2, 4 chimera. And here are the 2 inactivated vaccines. One is being developed by GlaxoSmithKline with a Brazilian institution. It's a formalin-activated 1, 2, 3, 4 viruses grown in tissue culture and inactivated with formalin. And Merck Sharp and Dohme has a genetically expressed subunit vaccine. And Sanofi is way out ahead. They're in phase 3. They have as many as nearly 40,000 individuals of all ages who've received the vaccine. Takeda is in phase 2 and is preparing to enter phase 3. The NIH vaccine has been licensed to various manufacturers. Butantan is a Brazilian manufacturer vaccine, and it's producing the NIH vaccine in GMP and entering phase 1 and planning for phase 2. The GSK and the Merck inactivated vaccines are both in phase 1. So what's happened? Well, the Sanofi vaccine has completed a clinical efficacy trial called a phase 2B, but there were 4,000 children in Thailand, and 3 doses of the vaccine in children, 50 to 60% of whom had already been infected with at least one dengue virus or had received a Japanese encephalitis vaccine. So this is a fairly immune population of children, 4,000. And after 3 doses of the Sanofi vaccine, 95 to 100% of the children had developed neutralizing antibody. But these antibodies, when the code was broken, did not protect against dengue 2, nor was the protection after 1 dose, 2 doses, and 3 doses, nor did that change. That was quite a surprise, no question. And here is the data. Here are the 4 viruses, the total number of years of exposure. And here are the vaccine, the cases that occurred in the vaccination and the cases that occurred in control. There were twice as many vaccinated children as controls. So the efficacy, you can see, against dengue 1 was about 60%, 80% against dengue 3, and 90% against dengue 4, but essentially no efficacy against dengue 2. Now the interesting thing is that after 1 dose, of course there were smaller numbers of cases, but you can see the efficacy essentially didn't change. Dengue 3 was pretty much the same all the way, dengue 4 was pretty much the same. So instead of cumulative increase, there was no increase. So both of those were great surprises. And I'm not trying to pick on Sanofi, but I think it's important. This is the first clinical vaccination experience that we've ever had in all the years I've worked on dengue, at least at large scale. And we need to try to understand what happened and what didn't happen with the Sanofi trial. But to do that, we first have to go back and remind ourselves of how does dengue immunity work in real life? How do dengue viruses produce immunity? Well, they actually produce 3 different types of immunity that we call homotypic immunity. Remember, there are 4 different dengue viruses. So if you've been infected with type 1 and challenge again with type 1, that's homotypic, that is highly protective. And then there's heterotypic immunity, and I'll talk more about this a little bit later, but if you're infected with 1 virus, you actually develop some level of protection against a second virus. And then there's what I call multitypic immunity, which means if you've had 2 different dengue infections, you're actually quite highly protected against a 3rd and a 4th. So most of the people in this room have probably had 3 or 4 different dengue infections. And if I asked you, most of you would, and I said, how many people in the room have had dengue hemorrhagic fever? So I'm not going to ask, but most rooms this size, nobody would raise their hand because dengue is very common. I'm sure that everybody living in Georgia has had dengue. And you've had 2 different dengues, and therefore you're essentially immune. You're not going to get dengue hemorrhagic fever. So we know something now from recent research studies that homotypic flavivirus immunity may be caused by an antibody that's been recently discovered. And this antibody interacts with the 3 different domains, domain 1, 2, and 3. And there's a cleft where these domains come together on the surface of the virion. And a similar antibody against West Nile has been discovered, and this is an image of what it looks like when an antibody is attached to the surface of the virus. So it turns out that the specific antibody, type 1, type 2, type 3, type 4, that probably mediates this type of homotypic immunity actually attaches to the outside of the virus. It doesn't attach to some specific protein on the surface. It attaches to a groove between each of the three proteins that make up the envelope. And here's a wonderful electron micrograph of this protein. This happens to be a dengue 1 human monoclonal antibody that's attaching to a dengue 1 virion. You can see it here. Here's the arm of the antibody, which is actually attaching to this joint. You see, here are the three different domains: yellow, blue, and red. So we've learned a lot recently about where dengue antibodies attach, but it turns out that antibodies may not be the whole story. For example, mice that have been immunized with a non-structural protein, which is not on the surface of the virus. In fact, non-structural proteins are only expressed when you're infected. But if you take one of the non-structural proteins, immunize mice, and then challenge them with dengue 2, they were able to resist challenge. And then another experiment, also in mice, showed that NS2A, NS4B, NS5, all of which were used to immunize mice, produced CD8 T cells, and they resisted dengue 2 challenge. So there may be a balance here between T cells and B cell immunity. So as I showed you, and you perhaps have read the article, there really was no protective immunity against dengue 2, despite the fact there should have been some cross protection between types 1, 3, and 4. So what actually happened? Well, one possibility is that when you mix all 4 viruses and stick them in one place in the arm, the multiple viruses may actually interfere and prevent processing. There may have actually been, the dengue 2 component may not have actually been protective for some reason. We'll talk about that in a second. And there apparently was absence of heterotypic or multitypic immunity. So let's look at these in more detail. Here's some early preclinical research done in monkeys showing that when all 4 viruses were given as a mixture, the monkeys developed neutralizing antibody just to 1 and 4. So you put them all together, and the monkey is only seeing part of the mixture of viruses. And we know that something like that happened in humans. And now here's a second dose, and you see you've improved a little bit. There's a little bit of antibody to dengue 3. Now here, in this particular experiment, they tried to get around this interference phenomenon by mixing the doses. They put 2 viruses in one arm and 2 viruses in another arm. So here's after a single vaccination. You can see there's a little bit more antibody to type 2, but still, this is predominantly a 1-4 response. And here's mixing other viruses, mixing up the viruses that are put in different arms, and it doesn't really change. When you get here to a third dose, a second dose, the antibody response is broader, and this is what Sanofi was hoping would happen. Is that the first dose might have only partially immunized, but a second dose would perhaps more fully immunize. But there's actually evidence from the preclinical monkey studies that the dengue 2 chimera by itself didn't prevent in disease, but it did prevent viremia. So here's a monkey that was immunized with dengue 2 chimera, the yellow fever, and here's a monkey that was immunized just with dengue 2 alone. And then these animals were all challenged with a wild-type dengue, and you can see that there's no viremia. Here are the control animals all showing viremia. But when the neutralizing antibody responses were measured in these and other monkeys, here's the post single one dose neutralizing antibody titer, and here's the challenge, the wild type virus challenge day 14 after the challenge. Look at how the antibody titers have boosted more than 8-fold. Look at some of these boosts. They're huge. Now what that is saying is that when you immunize an animal and then you challenge with a live virus, sticking a live virus in the arm, the antibody that's there should be good enough and strong enough to block the virus from infection. So you shouldn't have any infection at all. So this clearly shows that there was an infection. And this is a monkey. Monkeys are a little bit less susceptible to dengue than humans are. So there's a real possibility that the dengue 2 chimera itself, the dengue 2 chimera itself, didn't work. Now we don't know why. So based on the anamnestic antibody responses, these animals really weren't protected. So that's a major mystery. Now, the other major mystery is the fact that although we had almost 100% protection against dengue 4, we had 60% against dengue 3. If you give 1 dose and then 2 doses and 3 doses, the dengue 1 immunity ought to rub off. It ought to partially protect against dengue 2. Dengue 3 immunity ought to rub off and be partially protective against dengue 2. But that didn't happen. Now, we know from many studies over many years, Albert Sabin was the first to describe it, that if you infected somebody with dengue 1 and then tried to challenge them with dengue 2, within a matter of weeks, he couldn't even get them infected. But for 9 months, the disease was moderated. So there's a long-lasting cross-protection between dengue 1 and 2, and then many papers have been published showing many different viruses, dengue viruses, show some level of cross-protection against infection with a different dengue virus. So mild disease that occurs quite frequently. If the interval of 2 infections is very short, you're likely to have a very mild disease. If the interval is very long, you may have quite a severe disease. So one of the problems in dengue research and the way vaccines are monitored is that people use monkey kidney cells as a host for measuring neutralization. And that's the standard classical method. But if you switch over and use FC receptor cells, we know from lots of data I won't have time to review here, that dengue viruses after the mosquito bites actually propagate in monocytes and macrophages, and they go from monocytes to macrophages, dendritic cells to monocytes to macrophages all over the body. So the host cell that's supporting dengue infection and makes us all sick and that causes dengue hemorrhagic fever are monocytes and macrophages. And if you measure antibody in monocytes and macrophages, you get a different answer than if you measure antibody in monkey kidney cells. Here's an example of a study that was done some years ago where Bangkok school children, all of whom had secondary dengue 2 infections, of whom 7 were hospitalized, and 33 of these infections were fairly silent. Remember that a large majority of second dengue infections are actually silent. And what the author here did is she took, this is a cohort study where you bleed people before, these are school children, bleed them before they get infected, and then they get infected, and then you go back in the laboratory and you take the pre-infection blood and mix it with the virus that they were infected with to see what happens. So what she did was something that nobody has done since. She actually bled humans who were susceptible, didn't have any dengue antibody, and separated out their monocytes and put them in culture. And then when she she took the virus and the antibody from before the child got sick, and in this case she actually used undiluted serum, something that nobody ever heard of doing. But remember that in real life, neutralization or enhancement occurs in undiluted serum. It doesn't occur at 1 in 10, which is what we do in the laboratory. So undiluted serum plus dengue 2 virus, add it to human monocytes, and here are the results. The results were that virtually all of the children who had silent infections had enough cross-reactive heterotypic neutralizing antibody to protect against infecting monocytes. But the children who were hospitalized failed to protect. In fact, they actually enhanced. So they're actually circulating antibodies from the previous dengue infection, but at such a low titer that the antibody is able to enhance the infection rather than protect against it. So what do we know about heterotypic immunity? Well, it's very strong immediately after a dengue infection. That as time goes on, it persists, but it moderates the severity of disease. And it occurs for years and years and years. I did a study in Cuba many years ago where the interval between the first infection, dengue 1, and the second infection, dengue 2, was 20 years. And the disease was 40 times more severe than if the infection had occurred in an interval of 1 year. So as time goes on, you're losing this heterotypic immunity. And in fact, in Cuba, when these very low levels of heterotypic immunity were circulating, the attack rate of overt disease that occurred with each second dengue infection in the sequence 1, 2 was virtually 100%. We've never seen anything like that before. So heterotypic immunity may be partially mediated by antibodies that are better measured if we use Fc receptor bearing cells, but it may and they decline over time. But there really is evidence, and I don't have enough time to summarize it here, but it looks like heterotypic immunity itself is partly mediated by T cells. Now the final type of protection is what happens after you've had 2 different dengue infections. Well, what happens in real life is that, this is the Bangkok Children's Hospital study, 30 years of hospital experience. Very rarely did they see any children with third dengue infection. So that means that any combination of 2 different dengue virus infections protects you against a third. And this has been shown also in mice, where mice after 2 different dengue infections produce very cross-reactive but highly avid antibodies. And humans do the same thing. After 2 different dengue infections, you produce cross-reactive and highly avid dengue antibodies. So that may be important in protection. One of the things that I like to try to emphasize to people that are interested in dengue is that dengue occurs under 2 completely different circumstances. One of the only diseases in medicine that's anything like this. It's absolutely extraordinary. It was all discovered because there's a bimodal age distribution curve for children who are admitted to the hospital. This is a Bangkok Children's Hospital. And if you look at this first mode, that is infants less than 1, here's a Vietnamese infant with enlargement of the liver diagrammed on the abdomen. You notice this is a very healthy child. Severe dengue involves very healthy people. Yeah? You have to be very healthy to get in trouble with dengue virus. And here is the actual age distribution in the first year of life. You see that during the first 3 or 4 months, there's no dengue. The antibodies this is in Thailand, are protective, and then as they decline, they finally get to a point where optimally enhancing, and then that in turn falls off, and you begin to, by the end of the first year of life, children, at least in Thailand, are for the first time actually completely susceptible. This is all based on acquiring maternal antibody, and that maternal antibody at birth is maximum titer and is protective, and as it's catabolized at a half-life of about 40 days, you slowly lose it. Like it's a twofold serial dilution, 1 to 2, 1 to 4, 1 to 8, 1 to 16, and so on. And so you finally get through the neutralizing power of this antibody to the enhancing power, which lasts for about 6 months. And this is an example of the kind of antibodies that you get if you've had 2 different dengue infections. So cross-reactive, fairly high titers. If you did avidity determinations, these would be fairly avid. But we didn't see this cross protection with the Sanofi vaccine. Perhaps if the immune responses had been measured in Fc gamma receptor cells, I prefer primary human monocytes and macrophages, maybe we would have had a different answer. We won't know until Sanofi does the experiments. But we've talked about T-cell immunity. Now, one of the interesting things about Sanofi is that they have a chimera, and so all the non-structural antigens in this vaccine are yellow fever. So the structural antigens is dengue 1, 2, 3, and 4, but all the non-structural antigens are directed against yellow fever. And we know that yellow fever vaccine does not protect against dengue, so there's no cross-protection between yellow fever and dengue. The thing that's missing in the Sanofi vaccine is the ability to make T cells. So the Sanofi vaccine failure itself points to the importance of T cells. So monotypic immunity is most likely due to antibodies with T cells in secondary role. Heterotypic immunity there's a significant role for T cells with perhaps a secondary role for antibodies. And multitypic immunity, again, is a mixture of T cells and antibodies. So if we're going to make vaccines, we probably have to pay a lot more attention to T cells than we've paid in the past. So here are the 3 problems that any successful vaccine faces. One is it going to be possible, if you use adjuvants or one trick or another, to produce effective and long-lasting protection by presenting only dengue structural antigens? That's essentially what the Sanofi vaccine is doing. That's what the killed vaccines are doing. Or does protection require exposure to non-structural antigens, and must they be dengue? And if it is dengue, do you have to have each of the 4 dengue T cell epitopes, or is protection against one of the 4 dengue sufficient to protect against the other 3 viruses? So here's the implications I've been saying, is that Sanofi vaccine presents no dengue non-structural peptides. The Takeda vaccine presents only dengue 2 non-structural peptides, but not 1, 3, and 4. The NIH vaccine presents structural proteins against 1, 3, and 4, but not to 2. GSK, a killed vaccine, presents mostly structural, and Merck presents only structural. So all of that, big vaccine companies have really made an enormous gamble that structural proteins by themselves will be protective. So is there an end to the story? Are there answers to the questions that I've raised? Surely the questions will be answered. Time will tell. But for those who are waiting for a dengue vaccine tomorrow, I'll have to say, you're going to have patience. Thank you very much.